Good evening, everybody. My name is Jean Foucault. I'll be covering for several people today, and today we are going to be going over the Genetics Exam 2 ATA Review slideshow to try and help you prepare for the future exam. I'm still trying to study these things too, so please forgive me if I make any mistakes, but I'll try my best. Okay, so first let's start with gene interaction. We know that the way genes are expressed, their phenotypes, can be affected by the interaction between different genes. This can change our phenotype ratios to deviate from the Mendelian ratios, and it can sort of be categorized, like their interactions can be categorized in different manners or different phenotype things. We'll see when in the future. So what we're going to go over in terms of interactions are going to be epistasis interactions, incomplete dominance interactions, and codominance interactions. In addition, silent mutations can also be caused by gene interactions. Well, yes, there are several mechanisms for this, but we'll see later on. And we can also see that complementation tests can be used to determine the number and identities of genes that are causing a mutant phenotype to occur. Please remember that the dominance of one allele over another is also determined by protein products because they are eventually the end result of the DNA transcript, and they are what makes up the body and its cells and all of its proteins. Now, if we look at the chart to our left, this will represent mutant alleles recessive to the wild type. We can see that in the heterozygotic pair, the wild type and the recessive allele is still able to create enough enzyme activity that will reach or above the wild type phenotype threshold. This means that although there is a mutant allele, the genome is still able to create enough enzymes or create enough activity to maintain the wild type phenotype, which is why this allele is recessive and the phenotype at the end is going to be the wild type, right? Now, if we compare it to the one on the right, the mutant allele T2 is going to be dominant to the wild type T1. In this case, just one mutation of T1 into T2 is enough to decrease the enzymes and their activities below the wild type phenotype threshold over here, which will cause the mutant type which will cause the cell to exhibit the mutant phenotype with just one allele over here. Right? So that makes it dominant. Now, in some cases, the alteration or mutation of genes will cause the loss of some or all of its normal function. Hypomorphic loss of function mutations are those that loses only some of the normal function, while null loss of function mutations are going to be cases where there's going to be a loss of all of the normal functions, right? In addition, normal alleles can be defined as either haplosufficient or haploinsufficient, depending on how their phenotype is expressed or how they are expressed through phenotypes. For example, loss of function mutations are recessive if the normal allele is haplosufficient, and loss of function mutations are dominant if the normal allele is haploinsufficient. To the one to the left, we know that this heterozygote is going to create a normal phenotype, meaning that just one functional protein, or at least one set of the alleles of the normal alleles, is enough to make enough functional protein for the normal phenotype. Therefore, this is haplosufficient, and the loss of function mutation is recessive over here, and can only show up in homozygous recessive genotypes. Does that make sense? Somewhat. As for the one on the right, looking at the heterozygous genotype, we can see that it creates the mutant phenotype over here. Now, this functional protein, only one of them in the heterozygote, is not enough to create enough proteins for the normal phenotype, and as such, it is haploinsufficient. It's insufficient on its own. This mutation then is dominant over the functional protein, or this allele, and as such the loss of function mutation is dominant and the normal allele is haploinsufficient. Over here, arrow. Now, let's talk about codominance and incomplete dominance. Codominance is when the phenotype of both alleles are fully expressed in the heterozygote. Take these blood cells for example. The A genotype, or the A allele, the A allele encodes for the A antigens on the surface, while the B allele codes for the B antigens on the surface. If it is heterozygote between A and B, then the phenotype of both A and B will be fully expressed on the surface, a combination of A and B. Now, if we're looking into incomplete dominance, then neither of the alleles is completely dominant like over here. Instead, an intermediate phenotype is displayed, or a blend of both alleles is displayed. We can see this when we cross these homozygous red with these homozygous white flowers, or roses, which gives us these homozygous pink. We know this is incomplete dominance because the alleles, red and white, when blended together is pink, the phenotype that is displayed over here. This is the second filial generation over here, which is a hybrid cross between the red, or hybrid cross between the pink flowers, I think. And about a quarter of them is going to be homozygous red over here, pure red, homozygous white over here, and then half of them is going to be heterozygous for red and white. And so half of them is going to have the intermediate between the two, which is pink over here. This is a 1 to 2 to 1 ratio for both genotypes over here and the phenotypes. So for codominance, it means, okay, let's look further into the blood typing, right? So there are 3 alleles, O, B, and A, with O encoding for no antigens. That's why if you have both O and both O alleles over here, you're gonna get blood type O. But if you get the O allele combined with either A or B, it's going to create either A or B blood type over here. Because of that, we can sort of call the O allele recessive. Now, using this chart, what alleles are codominant? I'll give you a second. Second is up. And we can tell that the IA and then the IB alleles are codominant. Whenever they are in a heterozygous, or whenever they're together in a heterozygous genotype, both alleles are expressed independently and fully as antigens and in the blood type. Hello. Okay. Now, let's move into gene interactions plus, I'll take my notes out because this can be a bit confusing. We covered this line at the first, and then let's go ahead and cover complementation tests, right? So, let's say you have 2 mutants that are lacking a wild-type phenotype with one homozygous for the recessive mutant allele of one gene, and the other homozygous recessive for the mutant allele of another gene. If the wild-type phenotype is present in the offspring of the cross between these two, then the 2 genes, the 2 wild-type genes still remaining in each of the mutants, are going to complement each other. Both wild-type genes, unique to each mutant, provide the proteins that interact to produce the wild-type phenotype in the progeny. They complement each other. It can be a bit confusing, so yes, good luck with that. I recommend studying that a bit further. But it's basically showing that certain genotypes, when combined, should be able to create a phenotype that is needed. And let's move into duplicate gene interactions. These occur when 2 genes that encode for the same product are redundant. This means that they cover the same product, and as such, you only need one to be dominant in order to fulfill one phenotype. Okay. So there are 2 outcomes for this. The wild dominant phenotype is present because at least one gene has the correct allele, or the phenotype is gone because both genes lack the wild type allele. This occurs only when the genotype is homozygous recessive, which leads to the 15:1 ratio in this dihybrid cross. Let's move into the dominant gene interactions, the 96:1, the disc or sphere fruit, and there is a... Never mind. There's no charts for this one, but I'll explain it. So there's a 96:1 phenotype for dihybrid crosses for this ratio. The presence of one dominant allele of either of the two genes is enough to lead to a shared phenotype, which this represents the 6 of the ratio. But when both of the genes have a dominant allele, the two dominant genes interact, producing a new phenotype. If the organism is homozygous recessive for each, it will receive a unique phenotype which is only present in 1/16 of the progeny. Now let's move on to epistatic interactions. These occur when genes interact with one another in a shared pathway, and because of this, there is going to be some changes in the way phenotypes are expressed. Now let's look into recessive epistasis with the ratio of 9:3:4. This is when homozygosity for a recessive allele in one locus masks the expression at the second locus. Let's look over here. This can be seen in golden retrievers or the mice in this chart, right? So we can assume that the A gene affects the color of the fur, with the A dominant conferring brown fur, and a recessive A and the A recessive conferring black fur only in the homozygous recessive A organisms. However, C is a gene that affects whether or not the pigment is expressed. Only in C dominant allele are the pigments expressed, but if the organism is homozygous recessive for C, there will be no color at all, and this masks any type of expression conferred by the A allele. So as we can see over here, these homozygous recessive Cs masks the colors that should be over here. They should be black, they should be brown, but they're masked by the homozygous recessive. Let's move on to dominant epistasis. With a 12:3:1 ratio, the dominance of one allele masks or reduces the expression of either allele or gene. Let's look at the splash, for example. The presence of the B dominant allele confers a white color, which masks over the yellow color given by dominant A over here. We can tell that because there is a dominant A, and there is the yellow color over here and over here. Now, only when B is homozygous recessive can organisms with the dominant A allele show its yellow pigment over here, right? But if both genes are recessive, a green color is shown over here. Let's move on to dominant suppression. A dominant allele of one gene suppresses dominant allele of a second dominant allele, and it produces a 13:3 ratio. Now, let's go ahead and test your understanding. The question is, if a black and white true breeding mice are mated and the result is all gray offspring, what inheritance pattern would this be indicative of? It's indicative of incomplete dominance because the heterozygote mice is going to express a phenotype that is the intermediate of the white and the black alleles, which is gray. Hello. Okay. In case of codominance, the gene product is produced from both alleles. And here's an example with cows instead. We have a white-colored cow and a red-colored cow. They're both purebred, homozygous, and they create a hybrid cow, Rogan colored. And as we can see over here, the brown and the white are fully expressed but in different areas of the body, and as such, this is codominant. The gene product is produced from both alleles and expressed fully. Red and white. Now in the case of incomplete dominance, a monohybrid cross will result in an F2 with the 1:2:1 blink ratio. That's right. Both genotypes and phenotypes will be 1:2:1. This is because you'll have a quarter being homozygous red, homozygous white, and then half of them will be heterozygous, but because this is incomplete dominance, these are going to blend together to form pink, and so half of these are pink. There's one red, two pink, and then one white for one homozygous, and then two heterozygous in the middle. Let's move on to synteny, genetic linkage, and gene mapping. Now, synthetic genes are genes located on the same chromosome, while linked genes are synthetic genes that are so close together on the same chromosome that their alleles will not sort independently. It's important to note that all linked genes are synthetic, but not all synthetic genes are linked. As we can see over here, these two are linked because they're so close together, they are synthetic. These are not linked and they are not synthetic because they're on different chromosomes, but these two genes are synthetic but they are not linked because they're just so far apart. They will sort independently. Alleles of synthetic genes can be reshuffled when crossing over occurs between homologs to produce recombinant chromosomes. Crossing over occurs during prophase 1 and prevents linked genes from segregating together because they're just too close and can't be separated or are less likely to be separated. Each crossing over event, as seen over here, is going to create two recombinant chromosomes. As we can see, this crossover event occurs between A and B over here, creating two recombinant chromosomes once these chromosomes are detached from the other chromosome over here. Linked genes have a recombination frequency from 0 to 50%, but they do not surpass 50% as that is the maximum recombination frequency. There is complete genetic linkage in which no crossing over occurs, which is not as common, and then there's incomplete linkage where it mostly produces parental gametes but also recombinant gametes, which is the lower amount or in the lower quantity because it's less likely that the genes are going to be split up, recombined during crossover, and creating the recombinant gametes. Which pair of numbers here then represents the recombinant gametes? Yep, these are the ones that represents the recombinant gametes. Now in independent assortment, let me get my notes. In this chart, the genes exist on different chromosomes over here. As such, when you split the chromosome into the gametes and join the gametes over here, the genes are sorted independently from one another. There is a 50% chance the haploid cell will have parental gametes over here, which is pairings that are found within the parents over here. But there is also a 50% chance that the haploid cell will have recombinant gametes with a gamete from each parent from the swap, right? They are sorted independently, and as such, if you pick them out of a bag, one from each pair, 50%, 50%. When the genes are linked and on the same chromosome, however, you will produce more parental phenotypes and expected standard numbers than expected. The closer the genes, the lower the chance of crossing over, and subsequently the higher number of parentals over here. As you can see over here, D and E are so close together that they are linked and crossing over is less likely to occur. As such, when the gametes are joined and given the chance to cross over during prophase 1, it's more likely that the linked genes will remain together on the same gamete than it is that the genes are separated and a recombinant gamete is formed. This explains the pattern of parental and recombinant gamete frequencies in linked genes. What if you wanted to check whether or not these genes are linked? You'll have to do a test cross between a dihybrid organism and an organism with homozygous recessive alleles for both genes over here. And if there is a 1:1:1:1 ratio of the offspring for all possible phenotypes, we can say that the genes are independently assorted and the genes are unlinked, as in this case. 98, 103, 102, 197 are very close to the 1:1:1:1 ratio, and as such they are independent. However, when this dihybrid cross with the monozygote, however, when this test cross produces a offspring that does not follow the 1:1:1 ratio, we can assume that the genes are linked, which leads to the production of less recombinant phenotypes over here, and the production of more parental phenotypes as shown by 104 and 101, right? Now, in mapping the distance between genes, we can use centimorgans with capital M, and one centimorgan has a crossover frequency of 0.01. So instead of measuring the distance by unit of measurement, we measure it by the probability of crossover. As such, if we know the centimorgans, we multiply it by 0.01 in order to get the frequency of recombinance between two genes. In addition, the genes are additive. Now, how do we measure the recombination distance between two genes? Well, first, we have to test if the genes are linked or not using the 1:1:1:1 ratio. And then we have to find the recombination frequency, which is found on the bottom of the screen, the number of recombinants or non-parental progeny divided by the total number of progeny. Take this for instance. There's going to be a test cross between a dihybrid plant, TV plant, and another pea plant that is homozygous recessive for all of the alleles. Yellow is dominant over green, shown by the Y, and then round is dominant over wrinkled seeds in shape, shown by the R. Now we can tell from the get-go that these are linked genes because it does not follow the 1:1:1:1 ratio. 43 and 47 then has to be recombinant because they are less in frequency than these parental gametes. So we find these, we sum these two together, and then we divide it by the amount of progeny in total. And we will find out that about 9% of the progeny is going to be recombinant. As such, the genetic distance between Y and R genes is found by multiplying this frequency by 100, which leads us to 9 centimorgans, right? Let's go ahead and test your understanding. When genes are on different chromosomes, what percentage of gametes produced by a double heterozygous individual will be recombinant? 50%, because they are on different chromosomes, they are unlinked, and as such, the gametes that are recombinant will be 50%. There will be an equal amount of recombinant and parental gametes when it comes to unlinked genes. Let's say then that a species has n equals 4 chromosomes, and that the genes on the same chromosomes have a maximum recombination rate of should be 50%. If they're on the same chromosome, it doesn't mean that they have to be linked together, and as such, it can be treated as if they were independently assorted into the gametes, and as such, there's a 50% chance that there's going to be non-recombinant, and a 50% chance that it will be recombinant over here. Crossover is a 50-50. These are equal. The minimum recombination rate for the genes on the same chromosomes then will be 0%, because if they're on the same chromosome, then the ones that will have the minimum recombination rate are going to be with the ones that are completely linked together, meaning that they cannot have any crossovers as talked earlier. Let's talk about the partitioning of genetic variants and heritability. Now, genetic variance is represented by VG up in there. However, there are multiple types of variances. In additive variance, the variance is due to the effect of individual alleles. Looking at the example to our right, we notice that each instance of the dominant A allele increases the height by 10 starting from the homozygous recessive genotype. As such, the variance in height is caused by the additive effect each A allele gives on an individual basis. Now, in dominant variance, the dominance relationships among alleles itself will cause the variation. As we can see over here, the homozygous dominant and recessive genotypes have the same phenotypes as each other. But the heterozygous genotypes, there is a new phenotype of variant, if you may say. This is because there is a relationship between the dominant and the recessive that will cause the variant phenotype. Keep in mind that both of these variances could occur at the same time. In this example, the capital A allele instead adds 5 centimeters to the height, so when you have two homozygous A, you are at 20 centimeters, the addition of 5, 15, and then 20. Now, what about this weird anomaly? Well, we can explain this by saying that it is a heterozygote, and because of the dominance relationship, it is now 20 centimeters. This variance is explained through dominance. This variance is explained to additive, and as such, both are present in this genotype-phenotype variance. There is a third type of variance, interactive variance due to epistatic interactions between alleles. And when you add additive, dominant, and interactive variance, you will get genetic variance. The genetic variance is calculated first by determining the variance of the means among the lines in the last row of the table found over here on the bottom. You'll first find the differences between the means of the last row and the total mean, square them, and then find the average, as shown over here. In doing so, we quantify the variation of genotypes due to genetic reasons. We then calculate environmental variance through the same method, only this time using the means on the rightmost column, and this will quantify the amount of variance traits have due to environmental reasons. We add these to calculate the trait variance over here. Here is the population variance over here. The population mean found over here is going to be used over here, and then there's going to be something over here. This is going to be the general model over here. The summation of the squared of the variations, the average that, yes. This is going to be an example. We can see that there's genetic variance and environmental variance. Do these genetic and variance and environmental variance add up to get the population variance or the trait variance? They do. 2.67 times 2, 5.33 over here. We expand this equation over here, Vx, Vg, Ve, into these equations over here. When you're looking at the trait variance, you're going to be finding the average of the absolute variance found between the traits of the individuals by the traits or the mean of the population over here. So this is 62. Subtract that and square it to find the variance. Add that to the pile. Do the same thing for each of these tiny squares, and then you should get the trait variance. Genetic variance, you use the values found on the bottom over here. You subtract it by the total population mean, square that to make the variance absolute, find the average, and you should have the genetic variance. Same thing with environmental variance, only use the ones on the rightmost. Broad sense heritability. These measure the proportion of total phenotypic variation that is due to genetic variation. If the variation in a particular population is due to environmental sources only, and there is no genetic variation at all, then it'll be 0. But if all the variation in the population is due to genetic sources, then h squared is equal to 1. This is the proportion of the total phenotypic variation over here due to genetic variation. It's a fraction of how much the variation in genes affect the variation in phenotypic stuff over here. Narrow sense heritability measures the proportion of total phenotypic variation due to the additive genetic variation. So this time we're going to be dividing the additive genetic variation by the total phenotypic variation down below. This will measure how well a population will respond to selection. Let's talk about departures from Hardy-Weinberg equilibrium. There are several reasons why there will be departures from the equilibrium. There will be non-random mating, where individuals choose mates on the basis of heritable characteristics. The Wallin effect, which is a decrease in heterozygosity caused by varying genetic frequencies in subpopulations. Inbreeding, which is a form of non-random mating where individuals choose to reproduce with relatives. And finally, we will discover that the departure from the equilibrium will cause a decrease in heterozygosity and an increase in homozygosity. As we can see over here, there are many types of non-random mating. There's positive assortative mating, where there are bias towards individuals with similar phenotypic characteristics to the mate. Negative assortative mating, which is the opposite of that. You are, or at least the individuals, are attracted towards those with different phenotypic characteristics. Inbreeding, where the bias is towards mating with relatives, and outbreeding, which is biased against mating with relatives. As we can see over here, positive assortative mating is one of the causes for the reduction in heterozygosity. These are going to be the three most common matings in positive assortative mating, or at least the ones that are more boosted. There are the homozygotic breeding, which will produce only homozygotic offspring, and then the heterozygotic breeding, which will present only 50% heterozygotes for the next generation, while 50% are also homozygotes, which will just further snowball or compound and snowball into even more homozygotes offspring in these far corners. Keep in mind that due to positive assortative mating, homozygous will be less likely to mate with the heterozygous. Which of the following is not an assumption of HWE? I'll give you a second. Small populations is not an assumption of the Hardy-Weinberg equilibrium. Because small populations are more prone to genetic drift, that means some mutations can be fixated more, some alleles will be lost, or at least the genetic frequencies are going to be changed, thereby disrupting the equilibrium. Now, the Wallin effect is the consequence of genetic variance amongst subpopulations. This causes deviations from the Hardy-Weinberg equilibrium. It causes deviations in the heterozygosity for the population as a whole. The more different the gene frequencies among subpopulations, the greater the overall reduction of the heterozygotes, and this is a consequence of variance. Each subpopulation can still be in Hardy-Weinberg equilibrium, while the entire species will not be. There are two steps of calculating this. Step 1 is finding the heterozygosity, and step 2 is finding the fixation index. First off, you'll need to find the heterozygosity, the mean heterozygosity between the two subpopulations. You basically find the average of the frequency of heterozygotes present in the population to P1, Q1. The fixation index is going to use this heterozygosity, and it's going to subtract the average heterozygosity of the entire population by this heterozygosity, and then divide it by the heterozygosity of the entire population. This will help measure the reduction in mean heterozygosity compared to heterozygosity if it was in equilibrium. This is the effect of the heterozygosities of the different subpopulations. Let's look into this, for example. First, we'll look at the heterozygosity. The average of the heterozygote frequencies is going to be 0.18 plus 0.42 divided by 2, and then it's going to be 0.3 over here. Step 2 is the fixation index, right? So we are going to average these out to get this row. And then once we find P and Q over here, we're going to use these values over here as the values for the total population heterozygosity, which will be about 0.06. Now, let's go ahead and test your understanding. Let's say that in the population with n size equals 100, there's 100 individuals. The probability that a new mutation will reach fixation is blank. The initial frequency of the new mutation is going to be 1 over 2 over n, which is twice the population size because each member of the population will have 2 sets of genes, right? 1 divided by 200 is about 0.005, and there is a greater risk of it being lost, about 99.5%.